the study tried to enroll patients who have focal epilepsy and generalized epilepsy. Standard VNS has an indication, is approved for focal epilepsy, but has been widely used for all different types of epilepsy. So this one was, from the get-go, a slightly broader inclusion, and yet the numbers were small because this was a feasibility study looking at safety and efficacy, but really only is that really working. So the numbers are probably too small to be able to make a broad statement as to how well it works, but it was safe in terms of implanting it, and side effects during the stimulation were very similar to what you'd see with standard VNS, mainly an effect on voice. For these patients, in terms of seizure outcome, it was noted that within only a year, 12 months, there was a notable responder rate of over 60%, which means that over 60% experienced a seizure reduction by 50% or more. And of those, or of all comers, the seizure frequency reduction was 70%, meaning those that experienced a seizure frequency reduction had it reduced by 70% or more. That was a very good outcome, given that they were only followed for 12 months, and it is close to what has been seen with other neurostimulation devices after several years. It was well tolerated, as I mentioned, with no surprising or different or concerning side effects compared to standard VNS. At this point, the study was abandoned because of COVID. The pandemic made it basically COVID made it necessary to stop the study because people couldn't follow up or couldn't follow with a protocol, the scheduled appointments. So it is a little bit unclear what is going to happen with this particular stimulation setting. But I believe the company is thinking about using the findings so far and either expanding microburst stimulation treatment or re-exploring what is the effect really of the microburst stimulation settings and what is related to the individually titrated stimulation setting that is done with functional MRI. Meaning, the exciting part here is that functional MRI images were used to find settings that were thought or guessed to be particularly effective. And we don't have a biomarker that would tell you very quickly, like an image during, stimulation and fMRI, whether a person will have a good response with the stimulation setting. This is the first time that we have a visual biomarker that could maybe help us find settings that are particularly helpful to reduce seizures in a patient and have a better outcome altogether. So these are two different things that the study cannot answer, meaning how good is microburst stimulation compared to VNS, standard VNS, and how good or how important is using functional MRI to determine the best settings.